Imagine you're walking through a dense, remote forest. You see no animals, hear no calls. But what if you could take a single drop of rainwater that has dripped down a leaf, and from that one drop, know that a rare jaguar passed by hours ago? What if that drop could also tell you what the jaguar ate, the plants it brushed against, and even the insects that flew around it? This isn't a scene from a sci-fi movie. This is the incredible reality of a technology called environmental DNA, or eDNA. Welcome to G Fun Facts Online, where today we're uncovering the secrets ecosystems leave behind. So what is this magic-like substance? Every single living thing, from a blue whale to a bacterium to you and me, is constantly shedding its genetic material into the world. We slough off skin cells, we lose hair, we leave behind saliva and other waste. All of these bits and pieces contain our unique DNA, the fundamental instruction manual for life. When this genetic detritus is found in the environment, floating in the water, suspended in the air, or settled in the soil, scientists call it environmental DNA. Think of it as a constant stream of invisible, genetic breadcrumbs left behind by every creature. For centuries, our understanding of nature was limited by what we could physically see, catch, or photograph. Biologists would spend weeks dragging nets through the water, or setting up hundreds of camera traps, hoping to get a glimpse of their target species. These methods are valuable, but they have huge blind spots. They miss animals that are rare, shy, or active at odd hours. And they completely overlook the vast world of microscopic life. Our picture of nature was fundamentally incomplete. eDNA changes everything. By simply scooping up a bit of the environment, scientists can now identify which species have been in an area without ever seeing or disturbing them. The idea itself isn't brand new. Microbiologists have been studying the DNA of microorganisms directly from environmental samples for decades. But it was the revolution in powerful genetic sequencing technology in the early 2000s that truly unlocked its power for studying larger animals. A landmark study in 2008 used eDNA from a French pond to detect the invasive American bullfrog, proving just how incredibly sensitive this new tool could be. Since then, the field has exploded. How do you go from a scoop of mud or a bottle of water to a complete list of an ecosystem's residents? It's a four-step journey of high-tech detective work. First, you have to collect a sample. This is the field work part. Scientists might take a liter of water from a river, a core of sediment from a lakebed, or a handful of soil from the forest floor. The sample type depends on the ecosystem. Water is fantastic for aquatic life because it's a well-mixed soup of DNA from fish, amphibians, and invertebrates. Soil is more complex because DNA doesn't move as far, which can be a challenge, but it also allows for more precise location data. And scientists are getting incredibly creative. They're sampling snow from animal tracks, or even analyzing the blood inside leeches to see what animals they've recently bitten. And yes, that idea of using rainwater is very real. As rain falls through a forest canopy, it washes genetic material off leaves, branches, and the air itself, creating what's called rainwash eDNA. This gives us an unprecedented look into the often inaccessible treetop world of birds, insects, and even climbing mammals. Once you have your sample, step 2 is DNA extraction. This is a critical and tricky step back in the lab. Think of it as a needle in a haystack problem. You're trying to find picograms of target DNA, that's a trillionth of a gram, amidst grams of sediment, plant fibers, and other chemical compounds that can interfere with the analysis. The biggest challenge here is contamination. The method is so sensitive that a single stray skin cell from a lab technician could show up in the results. To prevent these false positives, labs have incredibly strict protocols, like working in sterile, DNA-free clean rooms and using dedicated equipment. Now you have your pure but tiny amount of DNA. For step 3, you need to make more of it. This is done using a technique you've probably heard of, PCR, or the polymerase chain reaction. It's the same technology used in forensics and medical testing. It's essentially a genetic copy machine that can turn a few strands of DNA into billions of copies, enough to be analyzed. Here, scientists can take two main paths. If they're looking for one specific species, like an endangered fish, they use a method called qPCR. It uses custom-built primers that will only bind to and copy the DNA of that one target species. It can even provide a rough estimate of how much of that DNA is present. But if they want to see everything in the sample, they use a powerful technique called metabarcoding. This uses universal primers that copy a specific gene region that is shared across a broad group of organisms, like all fish or all insects. This gene is like a universal product barcode at a supermarket. Every species has one, but each is slightly different. The final step, step 4, is sequencing and bioinformatics. This is where we read the story written in the DNA. The amplified DNA is fed into a high-throughput sequencing machine, which reads the genetic code of millions of DNA fragments at once. Then, the real detective work begins, using the tools of bioinformatics. Powerful computer programs sift through this mountain of data. First, they perform quality control, throwing out low-quality reads. They even hunt for chimeras, artificial sequences created when the PCR process accidentally sticks DNA from two different species together. Once the data is clean, the software compares each unique DNA sequence against massive global databases of known species, like the NCBI GenBank. Like a librarian cross-referencing millions of books, the software matches the unknown DNA from the sample to a known species, generating a detailed list of who was there. The result of all this work is a window into a hidden world, and the applications are already transforming how we study and protect our planet. Let's start with conservation. Finding rare and elusive animals is incredibly difficult. eDNA lets us know they're there without ever having to disturb them. For example, scientists can now detect endangered sea turtles just by testing scoops of sand from their nesting beaches. In the Mediterranean, researchers used eDNA from seawater to confirm that the critically endangered angel shark, which is rarely seen, is still surviving in the waters around Corsica, providing vital data for creating protected areas. And up in the Arctic, a project is testing ways to get polar bear DNA from their paw prints in the snow. This could offer a non-invasive way to identify individual bears, track their movements, and monitor their genetic health, all without the stress and danger of tranquilizing these massive predators. eDNA is also our new frontline defense against invasive species, acting as a crucial early warning system. In the United States, the tiny but destructive New Zealand mud snail has been spreading through freshwater ecosystems. Using eDNA, biologists found hidden populations in rivers where they were previously unknown, allowing for faster and more targeted control efforts. In another brilliant study, researchers used eDNA to create heat maps showing where an invasive fish was most concentrated in a river system. This allowed them to place their traps with surgical precision, dramatically improving their removal success and saving time and money. Perhaps most mind-bending is the use of ancient eDNA. DNA can survive for thousands or even hundreds of thousands of years in the right conditions, like in the cold, dark, oxygen-poor environment at the bottom of a lake, or frozen in permafrost. By drilling into the layers of lake sediment, scientists can analyze the eDNA in each layer, reconstructing the history of plant and animal life in that area over millennia. This has shown us how quickly ecosystems responded to past climate change events, giving us vital clues about our future. In the Arctic permafrost, They've found ancient DNA that tells the story of the lost ecosystem where woolly mammoths, steppe bison, and ancient horses once roamed. But maybe the most profound impact of eDNA is its ability to illuminate what scientists call the biological dark matter. We estimate that we have only discovered and named a tiny fraction of the species on Earth, with the vast majority being microscopic organisms like fungi, protists, and bacteria. eDNA is our telescope into this hidden universe. A recent study of deep-sea mud found that nearly 2/3 of the DNA they collected belonged to organisms that are completely unknown to science, organisms that couldn't be assigned to any known group. We are just beginning to grasp the true, staggering complexity of life, and eDNA is leading the charge. Now, as amazing as this technology sounds, eDNA isn't a silver bullet. It has its challenges. DNA degrades over time from sunlight and microbes, so a negative result doesn't always mean a species is truly absent. In a flowing river, DNA can travel far downstream, making it hard to pinpoint an animal's exact location. And as we mentioned, contamination is a constant risk that requires meticulous lab work to overcome. A major hurdle is moving from simple presence or absence to actual abundance. Detecting DNA proves a species was there, but it's still very difficult to know if that DNA came from one individual or 100. The rate at which organisms shed DNA can vary too much. Finally, our reference databases, while huge, are not complete. If a species' DNA hasn't been cataloged by scientists, we can't identify it from an eDNA sample. But the field is moving at lightning speed, and the future of eDNA is even more exciting. Researchers are developing portable, in-field devices that could give results in near real time, which would be revolutionary for managing invasive species threats or monitoring water quality on the spot. Scientists are also looking at environmental RNA or eRNA. RNA is much less stable than DNA, so detecting it would provide an immediate snapshot of which organisms are not just present, but alive and metabolically active right now. And the next frontier is functional genomics. This means moving beyond who is there. So to two, what are they doing? Basque. By analyzing the genes being expressed in an ecosystem, we could learn what metabolic processes are happening, or even detect the early stages of a disease outbreak. The potential for forensics is also being explored. Imagine being able to collect a suspect's DNA right out of the air at a crime scene. It sounds like science fiction, but the research is already underway. The story of environmental DNA is a powerful reminder that we are surrounded by an invisible world of information. The idea that a single drop of rain can hold the secrets of an entire forest canopy is not just a poetic metaphor. It is a new reality for science. It shows us that every part of our environment, no matter how small or seemingly insignificant, is teeming with the stories of life. As this incredible technology continues to develop, we will undoubtedly unlock even more of our planet's hidden secrets, giving us the knowledge we need to understand, appreciate, and protect it for generations to come. The eDNA revolution is just beginning, and it's changing our relationship with the natural world, one genetic fingerprint at a time. Thanks for listening to G Fun Facts Online.